And we're live.
Okay, so for number one, the first virus contains uracil, and so it's probably RNA. It's sensitive to endonuclease, but not exonuclease, so it's probably circular. And the proportions of the different of adenine and uracil and cytosine and guanine are close enough that it suggests that it's double-stranded. For virus number two, it also contains uracil, so it's also RNA. It's sensitive to both of the enzymes, so it's likely linear. And the proportions of adenine and uracil and cytosine and guanine are two different, so it suggests that it's single-stranded. For virus number three, it contains thymine, so it's DNA. It's sensitive to both the enzymes, so it's linear. And the proportions are similar enough that it suggests that it's double-stranded. For virus number four, it contains thymine, and so it's DNA. It's sensitive to endonuclease only, so it's circular. And the proportions are two different, so it suggests that it's single-stranded. For number two, DNA gyrase is composed of 4 protein subunits and uses ATP to compact bacterial chromosomes, while a nucleosome contains 146 to 147 base pairs and 8 histone proteins. Part B, the DNA double helix is 2 nanometers wide. Adjacent base pairs are separated by 0.34 nanometers. Part C, topoisomerase 1 introduces positive supercoiling. For D, the origin of replication in E. coli is ORIC. Part E, the sulfur is what labels the proteins, so 35S, and then phosphorus labels the DNA, so 32P. Part F, the sugar in uridine is ribose, and the sugar in deoxythymidine is deoxyribose.
All right, so for question 3, we're assuming that chromosome 14 and 21 are used completely, and so we add up their chromosomes. So we end up getting a chromosome length or a base pair length of a total of 157 million base pairs, and each base pair is 0.34 times 10 to the negative 9th apart. So timesing those two figures together, we get for part A 0.0534 meters. That's how long it is total. For the number of nucleosomes, we take 157 million, divide 50 plus 147. That's the number of base pairs that take up one nucleosome. And the answer is 801,020 nucleosomes. For part C, it's asking how many H2A molecules would you expect to find within that chromosome? And there are 2 H2A molecules for every nucleosome. So take 801,020 times 2, and that will give you 1,602,041 histone H2A molecules or proteins. For part D, it's asking about the number of radial loop domains. And we know that there are 25,000 to 200,000 base pairs per radial loop domain. So if we take both of those figures, we take 157 million base pairs for the entire chromosome, and you divide 25,000, that would give you 6,280. Take 157 million, divide 200,000, that will give you 785. And so we have a range of having between 785 and 6,280 radial loop domains.
Okay, so then for number 4, it talks about the bacterium, Mycobacterium tuberculosis, and how it has a singular circular chromosome that is 4,410,000 base pairs in length. So question A asks, if the chromosome was stretched out linearly, how long would it be in micrometers? So you just take the 4 million base pairs, and then we converted it to meters first and then into micrometers. So you multiply it by 0.34 and then by 10 to the power negative 9. And then you multiply that number by 10 to the 6 to get micrometers. And it ends up being 1,500 micrometers. And then for B, what you want to do to figure out the diameter is just take the 1,500 and divide it by pi, and you end up getting 477 micrometers as your diameter. For C, it's asking if one of the, it talks about if the genome is organized into a loop domain with each loop domain containing 50,000 base pairs. And then they're asking if one of these loops was stretched out linearly, how long would it be in micrometers? So you just do the same thing as you did in part A. So you just take the 50,000, multiply by 0.34, and then multiply that by 10 to the -9. But then you convert that into micrometers. So then you multiply that number by 10 to the 6th, and you get 17. And then question D, it's actually, we figured out it's a trick question. It's actually, there are 0 histones because this is a prokaryote, and prokaryotes don't have histones. And then question 5. So question A is asking which of the molecules are nucleosides, and the molecules that are nucleosides are A, E, and F. And then question B is asking which ones are nucleotides, and we said B, C, and D. And then C, they're asking which ones contain ribose, and we said C, E, and F. And then D, they're asking about which ones contain deoxyribose, and we said A, B, and D. And then E is asking which molecules contain pyrimidines, and we said C, D, E, and F.